So for our next talk, we have Linda Chio from the Gordian Biotechnology, more local, and she'll present her talk on multiplexed characterization of AAV cellular tropism and promoter expression in large animals using single-cell RNA sequencing. Linda?
Thanks for the introduction. Good afternoon, everyone. I'm really excited to share with you some of the work that we have done at Gordian Biotechnology. So first, to center ourselves for my talk, I'm going to first go through an introduction of the Gordian platform, where we are looking for cures for age-related diseases using a single-cell screening platform. And then afterwards, go through what this talk is mostly about how we have optimized our screens in order to look at curing these indications. And so first, my disclosures. So like I said, Gordian Biotechnology is a startup in San Francisco focused on curing age-related diseases. And how we do this is through pooled in vivo single-cell mosaic screens. And how this works is you might be familiar with barcoded serotype screens. Gordian does barcoded intervention screens where we take genes and do overexpression constructs or knockdown constructs to target these genes and barcode them. And so we have many, many genes that are packaged into an AAV library. This AAV library is then injected into an animal. We do all our screens in vivo, and this is where the term mosaic screening comes from, where we're doing the transduction at a low dose in which only a few cells are transduced with individual interventions, and each of these cells is now in the disease state of interest. We then isolate our transduced cells, and then running it through a single cell platform, we're able to look at the different genes that we're targeting and then compare the transcriptomic effects to see if there is now a return to a healthier phenotype. And the goal, of course, is to find a gene therapy in which we can cure these diseases. And so once we have a candidate gene target, we will then do a saturating dose within an animal and show the therapeutic effect. And so we are really interested in complex, non-monogenic diseases because Gordian believes that the next frontier of gene therapy is really looking at these non-monogenic diseases. And so we have 4 lead indications: osteoarthritis, non-alcoholic steatohepatitis, heart failure, as well as pulmonary fibrosis. As you can see, these indications are in very diverse tissue types. And why we're interested in the single-cell aspect of this is because within all these complex diseases, there are actually several cell types that are important for the progression of this disease. And in order to cure these diseases, we're going to have to figure out what is going to be therapeutic. And so, for example, for OA, you see that there's chondrocytes, which is the main cell type in the cartilage, as well as synovial cells, the other main cell type in the synovium. And what is healthy for a chondrocyte is not necessarily what is going to be healthy for a synovial cell. And so we run all of our screens and hopefully eventually our most desirable animal model to really get information that can translate to the human disease. And so, like I said, it's really important to look at the single-cell aspect of this and to really be able to get the best screen possible. And so we've done a lot of work using a single-cell screening platform to really come up with the most ideal serotype as well as construct to run our screens. And so, similarly, we do barcoded screens now for serotypes as well as promoters. How this works is we have pooled barcoded serotypes as well as pooled barcoded promoters. And so our constructs consist of a promoter as well as a reporter gene. This reporter is often a fluorescent protein that's being expressed as an RNA transcript. Additionally, we also have a barcode that's also being expressed as a small RNA transcript. We pool these constructs and inject them into our animal models. After 4 weeks of expression, we only collect cells that have functional expression, and then from there, isolate them using 10x genomics. We process these samples for our transcripts and our barcodes, and run it through next-generation sequencing to obtain our data. And from there, we process our data for our best hits. So we can look at specific cell types within the population that we've isolated and look at which vectors have ended up in these particular cell types with their barcodes. So first, as a control, we validated our barcoded screens in mouse cardiomyocytes for our heart failure program. We started with 17 barcoded serotypes and injected it into mice. What I'm showing you here is a UMAP. And so what this is a cell type classification in which we look at the transcriptome of the cells that we have isolated and then cluster them according to similarities to their transcriptome and looking at cell type annotations, identify what these populations are. So not surprisingly, within the mouse heart, most of the cells that we've isolated are cardiomyocytes. We look at the transduced cardiomyocytes and then verify the barcode detection within them. And what we see here in this screen is that looking at many different serotypes, we are able to replicate what was already shown in literature, where AAV9, MyoAAV2A, as well as AAV-Myo are really good at transducing cardiomyocytes. Next, we validated this using individual serotypes, where we were able to put AAV9 as well as AAV-Myo to cardiomyocyte tropic serotypes and show that they transduce very well in individual mice. And we also show that AAV2, which was not very good in our screen, is also, once again, not very good in an individual animal. We also validated this using histology. So next, we wanted to enable our pulmonary fibrosis program. In that case, there's actually no serotype in literature that can target lung fibroblasts. And so we're really interested in developing a serotype or identifying a serotype that can do so. So we started with a 16 barcoded serotype pool, and we injected it in both healthy and fibrotic mice. In the case of a mouse lung, this is for the fibrotic animal, you can actually see that the fibroblasts, that green population, is only a small proportion of the total cells. And so we're able to isolate the fibroblast population and then running it through our single cell platform, we once again looked at just transduced fibroblasts and look at which barcodes were detected. So compared to AAV2 and AAV6, which are known lung targeting serotypes, we show that there are several serotypes that can now transduce lung fibroblasts. Taking, once again, individual serotypes and injecting them into animals. What I'm showing here in the graph is on the x-axis, the percent fibroblasts transduced out of the total fibroblast population that we enriched for, as well as what percentage of fibroblasts are in all transduced cells using these particular serotypes, kind of denoting the specificity of these serotypes. And so moving forward for our mosaic screens in pulmonary fibrosis, we decided to use serotype L, which shows high specificity as well as the ability to transduce these lung fibroblasts. Additionally, we looked at histology and we show that the signal from the AAV, the green that you see on your screen, also co-localizes with two fibroblast markers, alpha SMA as well as PDGFR alpha. So now that we have the serotype that can target lung fibroblasts, we really want to also optimize the construct in which we run our screens. And so starting with a 44 combination construct pool, where our construct now has a different combination of enhancer, promoter, intron, regulatory elements such as WPRE, as well as poly-A, we can compare all of these constructs in a single or a group of animals, where you can compare things like number of transcripts per cell. Additionally, a challenge in using AAV is the amount of packaging space within the capsid. And so we can compare certain things such as the non-packaging space and the number of transcripts per cell, as well as the number of cells that we can isolate. And so compare these to CMV. We once again took individual constructs and show that looking at these two criteria, we've identified several constructs that are better than CMV. So like I showed, we can look at many, many things in a single animal and compare them pretty well. And so we wanted to do this with also our large animal studies. And so in order to really get an osteoarthritic screen going, we also need to find the optimal serotype for our screens in a large animal model, osteoarthritic horses. And so here we started with 13 barcoded serotype library. We injected into 4 joints. We looked at the transduction and the detection in synovial cells and chondrocytes, our 2 main cell types, and showed that compared to AAV2 as well as 2.5, 2 serotypes that are used in clinical trials for OA, that we have several serotypes that are more specific and highly transducing in these cell types. So for example, serotype A is highly specific in synovial cells, and serotype G is specific in chondrocytes. We then packaged a intervention screen within our 2 specific serotypes, and then showed through FACS transduction data that there is high specificity in the synovium as well as the cartilage through the fluorescence. Our single-cell data also corroborates that, where we show that serotype A is now mostly in synovial cells and serotype G is in the chondrocytes. And so for the results of this screen, I hope you stay tuned as we show what these interventions do in our animals. And finally, to enable our NASH studies, we looked at serotype and promoters in the NHP liver. Here we pulled 6 bar-coded serotypes as well as 3 bar-coded promoters and injected it into one adult African green monkey. We show that there is expected immune response to the foreign XFPs that we've injected. But even at the end of it, in our histology results, we show that there is still transduction, and we're able to show that there's several serotypes that can transduce these hepatocytes, as well as our promoters that were engineered also transduce these hepatocytes. So in summary, I just want to show the progress that Gordian has made in enabling screens in all 4 indications, and then hopefully in the future we will show the results of our intervention screens. And so with that, I'd like to just acknowledge everyone at our company. We're still growing. We are currently 30 people. And a big thanks to our co-founders, Francisco and Martin, and then also our collaborators in the University of Florida, as well as Wake Forest. We're a really fun group where we have monthly happy hours with the greater community, so if you want to hit us up, please do. And with that, I'm happy to take questions, and if you want to come speak with us, we have several posters as well.
Hi, I had a question about the design of your barcoded constructs. So you mentioned that the barcode was expressed from a separate promoter downstream of the poly A, presumably a polymerase III promoter, I would imagine, for compact aspect of it.
Yeah, so we do have expressed barcodes with small RNAs.
But aren't you worried that the expression pattern from these short promoters might be different from the ones of the larger POL2 promoters that are used to drive the rest of the construct? So you might have a different picture of what happens in terms of expression with those.
That's a great observation. We actually also have a fluorescent a fluorescent gene that we express as well. So these larger RNAs are also functionally expressed in the cells that we collect. So there are differences, but we are pretty sure that we have the effects of both.
And one last detail, the reason why you wouldn't put the barcode before the poly A in the major transcript is because of the ease of detection that is conferred by the stronger POL3.
There is definitely an advantage of using a smaller RNA transcript, and that's expressed very well.
Hi, nice talk. I wanted to ask about in your large animal studies, whether you pre-screen for neutralizing antibodies against each of your different serotypes. Because there's an abundance of literature that shows it's really hard to find animals that are naive of 1, maybe 2, let alone 6, 10, 16 different serotypes. And obviously these can influence your biodistribution, et cetera. You only showed 1 NHP there, and obviously you get variability from animal to animal depending on this. How are you teasing this out in terms of how it's affecting the biodistribution?
Yeah, so those are great questions and stuff that we always have in the back of our minds when we're running our screens. And so for the first question, when we're doing our barcoded screens, we actually do not look at neutralizing antibodies, but once we isolate a serotype and use it for our further intervention screens, we definitely look at the neutralizing antibodies there. We also, in most of the cases, other than that NHP experiment, we have several animals that we use, and so we kind of make sure that all the results are consistent with each other.
Thank you.
Hello, that was a nice talk. I'm Abhishek from Weill Cornell Medicine. So I had a question, like when the first screening when you do to understand like which is serotype would be good for transducing a particular cell type, but later on when you say do addition of promoters or say introns, did you notice a change in the percentage of the cells which were transduced before, like doing it on the second step of the cassette design? Like, does it change the population?
Are you asking if we definitely normalize for we want to have a minimum number of cells collected before we can do statistical analysis to make sure that the results of our screen is robust?
Yeah. No, my question was when you do that in the second step, do you see a change in the number of cells which is transduced? like say even for a particular serotype?
So that would depend on the dose that we use. I would say that I don't think that has been noticeable, but we do use different doses in our validating screens where we do a more saturating dose as compared to in our screen where there's many pooled constructs, and so there's a lower dose initially.
Okay, thank you.
Thank you.
All right, thank you very much, speaker.